Hey, thanks so much. An honor to be here and present. It's also, I noticed the Ides of March today. So, friends, Romans, husky men, lend me your ears. First, some disclosures. I want to start with the observation that for the first time in human history, we're actually able to grow new tissues from our own bodies that could typically only exist during embryogenesis. The way this works is that we can cultivate cells from any individual, grow them out. These are not stem cells, but we can reprogram them into pluripotent stem cells. These are called iPS cells. And these are a very early stage of embryonic development before any of the organs have formed. And we can then coax these cells to differentiate into complex tissue-like structures called organoids and use these in the lab for disease modeling. We can also put these structures back into the bodies of the original patient from which they were derived. And in this setting, you would have an autologous transplant that would not require immunosuppression and could be generated on demand. The reason I'm interested in this, and the kidney in general, is because of this man, my uncle, shown here after his second kidney transplant. And he lived with kidney disease for 50 years and only recently passed away due to complications. And I would love to see a future in which people like him don't have to suffer throughout their lives just to survive. So the outline of today's talk, my objectives are twofold. One is to talk about some of the highlights from our laboratory recently modeling kidney disease in a dish using these mini kidney organoids. And second, to update on where the frontier is for kidney regenerative medicine in our experiments. So a number of years ago, we made a really astounding discovery, which is that we can model polycystic kidney disease in a Petri dish. And this is what it looks like. This is what PKD looks like in a dish on the left. These are mini kidney organoids that have a mutation that causes the loss of function of either polycystin 1 or polycystin 2. And as a result, they swell up and form these balloon-like structures, very similar to the disease phenotype in vivo. And we know this is a specific phenotype to the disorder because if you look at an isogenic control, which is the same exact cell line, the only difference is the single gene has not been mutated, those don't form cysts. And this is highly robust and creates a phenotype that we can now mine to understand the disorder and develop drugs. And fundamentally, it teaches us that PKD cyst formation is a very simple process that we can somehow recreate with just cells in a Petri dish. You don't need an entire organism, you don't need flow, you don't need a vasculature. There's something about the cells. Now, the mutations we started off with in this system were insertion and deletion mutations. And we introduced these using the conventional CRISPR-Cas9 gene editing system. And although these create knockout mutations, in the clinical setting, these are almost vanishingly rare. You can see them here in this orange wedge. They are a very rare type of mutation. It's an artificial type of mutation. And the really common mutations are actually single base pair mutations in either of these genes, PKD1 or PKD2, that cause a stop codon prematurely in the expression of the protein. And these truncating mutations due to nonsense, they're called nonsense mutations because they stop the creation of sense amino acid residues. These are not only very frequent in PKD, but also predominantly cause a more severe disease phenotype. So these are the types of patients that tend to need treatment. So we would like to be able to model these disease mutations. And the reason for that is because there's a class of molecules called glycosides. And these glycosides localize to kidneys, and they can read through premature stop codons. So they trick the ribosome into synthesizing a full-length protein instead of the premature stopped protein, which is non-functional. So these are exciting therapeutics potentially for PKD. But the problem is that existing animal models of polycystic kidney disease also don't have these types of nonsense mutations that are typically found in the patient. So we have no way of testing these therapies in a preclinical model. So to address this, we envisioned using a new technology called CRISPR cytosine base editing. And the way this works is instead of just severing DNA and then waiting for it to repair itself, it tethers the gene scanning enzyme to a deaminase, which changes a cytosine DNA nucleotide into a thymidine. And this, if introduced in frame, will create a nonsense mutation, an early truncation. So this is a relatively new type of CRISPR technology, but it will allow us to create these types of mutations. And we identified 4 different types of mutations that are common both in PKD1 and in PKD2, and created these mutations using cytosine base editing. And this work was performed by a MD PhD student who's recently graduated named Courtney Vichy. And what Courtney did was express this base editor in either PKD1 or PKD2, targeting either of these genes. And as you can see, this generated a host of individual cell lines that either had the wild type, that would mean that they're unedited sequence, a heterozygous sequence where only one allele was replaced with a T, or a homozygous mutant where both alleles were replaced by T. And these are just a pictograph of the types of cell lines that Courtney was able to generate. We generated over 100 cell lines in this one experiment. So now the question, of course, is, do these get cysts? Just like our original CRISPR-Cas9, and indeed they do. And you can see this here in the water control from day 0 to day 14. And in each of our mutants, we have robust cystogenesis. This is also quantified below, so you can see how the cyst area is very high just in water. And when we apply these read-through glycoside therapeutics, they dramatically reduce cystogenesis for each of these mutations. So you can see here, as you titer in what's called ELOX-02, and we also did a similar experiment with another drug called ELOX-10, we see a reduction in the area of these cysts, and we also see reduction in the number of cysts, which I'm not showing here. So this was a very exciting result. This is really the first proof of principle for this class of drugs that these could be useful in reversing the phenotype of polycystic kidney disease. Now, you might say, well, this could just be a toxicity effect. So we tested that, and we did it a couple of different ways. So as we titer in these compounds, we performed both a live dead assay, and in this picture here, the green represents live cells and the red represents dead cells. We only see gross toxicity at around 300 micromolar of the drug, whereas at lower concentrations where we were seeing efficacy, we don't see these types of toxic effects. And similarly, we used a lactate dehydrogenase assay to essentially measure the release of an enzyme, which would only be released during cytotoxic events when cells lyse. And we saw no really detectable toxicity at the clinically effective, or we would say, organoid effective doses of the drug. So finally, we've got some evidence of safety. We've got evidence of efficacy. Do we have evidence that the drug is doing what we want it to do mechanistically? And this is really the triumvirate, I think, that you want to achieve whenever you're looking at any type of therapeutic development. So to measure the potency of the compound, we titered it in and we looked at the expression of polycystin-1 protein. And what we observed is that as you titer it in, we see an increase in polycystin-1 by Western blot. And this is detectable not only in our PKD1, but also our PKD2 knockout organoids, where polycystin-1 is typically degraded because it requires polycystin-2 for stabilization. And this was quantifiable. So we could see that the fold change in polycystin-1 was increasing up to about the wild type levels in each of our mutants when we titered in ELX01 or ELX02. So these data collectively show that we can get target engagement, safety, and efficacy. And this paper, which Courtney was working on along with other members of our team, is now accepted for publication. So the first conclusion that I have is that all models are wrong, but some are useful. We can recognize that these are not perfect, but these phenotypic models have the potential to reveal key ingredients of disease and let us do experiments relatively quickly that we can't do in any other type of model. And we now have a 5-year project together with Dr. Himmelfarb to establish mini kidney drug development tools along these lines. Can we get these to a point where the FDA, for example, would recognize them as an alternative to an animal model? And we believe that this is possible because this is the direction that the government has been going in. And quoted here is the FDA Modernization Act 2.0, which allows an applicant for market approval for a new drug to use methods other than animal testing. And this includes cell-based assays, organ chips, microphysiological systems. These are all the types of things that we're working on. And in general, these organoids fit in at different stages of drug development. So it's not only at the discovery stage, which is sort of where we are at, but you can envision now using this assay for lead optimization to make a drug that's going to be even better, more specific for the kidneys or more specific to the type of disease phenotype you're looking at, measure safety and efficacy preclinically and figure out how those doses correlate. And even when you're at the stage of clinical trials, there may be utility for having organoids as a system to investigate mechanisms that arise. And hopefully that in the long run, this will lead to more FDA approvals for therapeutics for kidney disease, which we all recognize is a great need. A future direction here is to branch out beyond polycystic kidney disease, and we've already begun to do this in a couple of major areas. So we're interested in modeling acquired disorders, and these include viral infections. We have a couple of papers on how SARS-CoV-2 infects proximal tubules in organoids. And we've also have some papers on APOL1 disease, which is a difficult one to model, but of great interest to us to understand how that disorder interacts with the immune system in specific patients and creates an FSGS essentially. We're also now beginning to model diabetic kidney disease. Of course, there's no single gene that you can mutate to cause DKD, but we have identified a preliminary phenotype in which high glucose can trigger changes in the podocyte population. And that's a paper that we are now trying to get published. So now I would like to move on to the regenerative medicine part of the talk. And I'll preface by saying that for as long as we've had these organoids, we've tried to implant them into mice and see whether they can engraft. So there's a long-standing effort on my lab's part, as well as other labs, to understand how well these structures can function or engraft in a living animal. And these are some images of the most recent publication that we had on this topic, where what we observe is that these are human cells. These are the green cells. They are implanted, in this case, beneath the kidney capsule. And we have human podocytes expressing nephrin, forming a little ring structure. And there's a small amount of vascular cells, and these are coming from the mouse, that are inside that structure. So this is a very primitive type of glomerular architecture that's arising. And this was exciting on the one hand, because we're getting some cells surviving, but it was not at the level where we would say this is really engrafting productively to form glomeruli. So there's been several studies over the years that support this idea that organoids, when you implant them into an animal, can engraft to some degree, that the tubules can survive, and that there is some level of vascularization that can occur. So I think that this is an ongoing field and is of great interest to the entire world, because if we could get these structures to engraft productively and create new nephrons, that would create functional tissue for people who need it and stave off the need for renal replacement therapy. So Thomas Vincent in our laboratory has taken on this project, and he asked a couple of simple questions. One is, can an organoid differentiate inside a mouse? So far what we've done in the past is take mature organoids and implant them into the animals. But what would happen if we took cells that were not fully differentiated before they turn into complete organoids and implant them? And then the other related question is, does it matter how old the organoid is at the time of implantation? And this was work that we did collaboratively in a project with Benjamin Deckel, who's in Israel and has a lot of expertise in stem cell differentiation. And one of the first things that we observed is that young organoids mimic nephrogenesis. So if you look at the early stages of organoid formation in the Petri dish, we observe the formation of these renal vesicle-like structures which express high levels of PAX2 and form these little rosettes. And these are analogous to developmental kidney structures that are normally found as the nephron matures in vivo. And then we see this wave of differentiation where the proximal tubule marker here, LTL in red, begins to form on one side of these. And you essentially have a differentiation of a neocortex beneath these renal vesicle structures, similar to what you have in vivo when the organ is forming. So now Thomas took these cells at that earlier stage, before they had started to really fully differentiate, and implanted them beneath the kidney capsule. And this is an image here of how this works. We have a mouse. The mouse is fine. He's just anesthetized. And we put a little tiny piece of tubing and simply implant the cells beneath the kidney capsule without doing any damage to the rest of the kidney. And what happens under these circumstances is quite remarkable. So the mouse kidney sends forth endothelial cells over a process of about 3 weeks. And these hook up with the podocytes, the human podocytes that are differentiating in the graft and invade them. And what we observe here is the mouse endothelium here is in green and the podocytes are in red. What we observe is this complex network of vasculature just inside this capsule of podocytes. And this is quite different from what we would get, for example, in a kidney organoid in a Petri dish, where the podocytes just form a little ball that has no vasculature associated. So how is this working? How are we getting such complex structures? We looked at whether there were other types of cells besides podocytes that were differentiating in our organoid grafts. And we observed that indeed, not only are we have human podocytes here wrapping around the endothelium, but we also have in red parietal endothelial cells, which are forming a capsule surrounding the entire structure. And interestingly, there are mesangial cells, which we identify here in the red. And these are them. They're purple because they're red and blue together, but these are inside the capillaries on the other side of the podocytes. So they're forming a mesangial bed, and this is probably necessary for the ability of these endothelial cells to properly invade and create what looks like a glomerular tuft. And it's also rather remarkable that the endothelial cells from the mouse have the ability to do this with human mesangial cells and podocytes and all the other types of progenitor cells that are present. So the second conclusion is that timing is everything. Organoid progenitor cells can indeed differentiate inside a living host. And we observed that these grafts can recruit endothelial cells to form glomerulus-like structures. Now, I know you're wondering, are these functional or not? And that's something we don't know. It's still rather early days, so we're just limited to looking at the structure at this point. But in the future, if we can get this to be efficient, we can envision creating a whole bunch of new nephrons for the host that would be plugged into the vascular system. And one of the future directions that we're working on is creating humanizable hosts that have kidney diseases. And this is one example. So these are mice that they have to be immunodeficient, otherwise they would not tolerate human cells. So these are mice that we've created on an immunodeficient background that have mutations in CTNS, which are the cause of cystinosis in humans. And we have a grant now to implant these types of cells inside the cystinotic mice and see if they can rescue any of the function that is missing in these animals. So all this talk about engraftment makes me also recognize the importance of growth niches. And it's really about the environment in which cells find themselves that allows them to productively succeed. And that is also true of human beings. So I wanted to take a moment just to recognize some of the growth niches that I've been lucky to have. When I was a postdoc, I had a really great group of mentors, including Dr. Joe Bonmentry, first and foremost. And he taught me the fundamentals of the kidney and took great risks in investing in me at a time when this whole field didn't even exist. And then when I came to the UW, I was very fortunate to have the mentorship of two individuals, Stuart Shankland, who's really been helping with the regenerative side of this work and bringing me into his fold on the glomerular angle, as well as Jonathan Himmelfarb, who's been an essential partner in crime for me on the microphysiological systems and just in general here at the UW. So I wanted to take a moment to acknowledge that and particularly thank Jonathan for his contributions over the years. So with that, I'll conclude. I wanted to thank everybody in the lab who's been involved in these projects. Thomas is here, and you can ask him about his graft experiments. We've had a great group of alumni who have gone on to great things. And this is the lab pictured here from a recent photo. And the different funding organizations, it really has taken a huge village to get this type of work funded and done, and to the point where it can be published. We really are 100% reliant on these types of grants from different organizations to grow the lab. So I thank them as well. And I thank you for your attention.